Assalamu alaikum wa rahmatullahi ta'ala wa barakatuh, and welcome everybody. This is my first video on my YouTube channel. If you like it, please like, subscribe and comment to receive more videos in the future. Let's start our video today. Here we go.
الرحمن الرحيم والحمد لله وكفى والصلاة والسلام على الحبيب المصطفى رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقه قولي. السلام عليكم ورحمة الله تعالى وبركاته. In this lecture we will talk about glycolysis. This lecture is presented by Elham Omer, MSc Medical Biochemistry, Khartoum University. Let's see the objectives of this lecture first. By the end of this lecture, all the students should be able to define glycolysis, identify the general properties of glycolysis, discuss the biomedical importance of glycolysis, describe the steps of glycolysis, differentiate between glucokinase and hexokinase, explain glycolysis in RBCs, distinguish between energy produced under aerobic and anaerobic glycolysis, and recognize the effect of pyruvate in glycolysis. Let's explain the term glycolysis. Glyco means glucose and lysis means breakdown. So it is a pathway concerned with catabolism or breakdown of glucose. ده مسار تكسير الجلوكوز. The major pathway for utilization of glucose so as to produce energy. It defined as series of enzymatic reactions which convert glucose into pyruvate with concomitant release of energy in the form of ATP or an reducing equivalent in the form of NADH. هو عبارة عن سلسلة من التفاعلات الإنزيمية بتحول الجلوكوز لمركب يسمى البيروفيت. Through this process there is the production of energy either in the form of ATP or reducing equivalents in the form of NADH. ATP referred to adenosine triphosphate and this is the currency of energy. دي العملة اللي بنتعامل بيها مع الطاقة. نحن ما بنتعامل مع اليورو ولا الدولار ولا الجنيه السوداني. بنتعامل مع ال ATP. How many ATP molecule produce and how many ATP molecule consume. NADH nicotinamide adenine dinucleotide and this through the respiratory chain produce 2.5 ATP. Later on لما نتكلم عن ال respiratory chain بنعرف how the NADH produce 2.5 ATP. General properties of glycolysis. Number one, it is the universal pathway. That means it occurs in all tissues. Universal يعني عالمية بتحصل في كل التشوز. RBCs, brain, adipose tissue, whatever. It is a cytosolic pathway. That means it occurs in the cytosol of the cell. لازم نعرف أي pathway in which compartment it occurs. Is it occur in the cytosol or in the mitochondria? حتى في الميتوكوندريا is it occur in the inner mitochondrial membrane or in the matrix. It is a unique pathway since it can occur in the presence of oxygen aerobic or in the total absence of oxygen anaerobic. Glycolysis فريدة في نوعها ممكن تحصل في وجود الأكسجين أقول عليها هوائية أو في عدم وجود الأكسجين لا هوائية. In both cases reactions are similar except in the end product and extent. Under aerobic condition the end product is pyruvate and under anaerobic condition the end product is lactate. الاستمرارية. Under aerobic condition pyruvate will be converted into acetyl-CoA which enter Krebs cycle for complete oxidation into carbon dioxide and water. And under anaerobic condition pyruvate is converted into lactate. Biomedical importance. Glycolysis is the principal route for glucose metabolism leading to the production of acetyl-CoA which enter Krebs cycle for complete oxidation into carbon dioxide and water. And the main objective of this pathway is the production of energy. It is the main pathway for metabolism of fructose and galactose. Also, the other hexoses like fructose and galactose can be metabolized by this pathway. Production of ATP in the absence of oxygen. This allows skeletal muscle to perform at very high level when aerobic oxidation becomes insufficient. When we talk about the properties of glycolysis, we said that it can occur under aerobic and anaerobic condition. Muscle during exercise, where there is a short of oxygen, glycolysis can occur anaerobically and produce energy. Diseases associated with glycolysis include hemolytic anemia due to deficiency of pyruvate kinase. This enzyme catalyzes the last step of glycolysis. When this enzyme is deficient, lead to hemolytic anemia because there is a short of energy, and RBCs it has no enough energy, and this will cause hemolysis of RBCs. Fatigue due to deficiency of phosphofructokinase 1. This is the key enzyme of glycolysis. When this enzyme is deficient, lead to fatigue, الشعور بالإرهاق والتعب لأنه ما في enough energy. Lactic acidosis due to deficiency of pyruvate dehydrogenase complex. This enzyme converts pyruvate into acetyl-CoA. When this enzyme is deficient, pyruvate will be converted into lactate, and the lactate will accumulate and cause lactic acidosis. Steps of glycolysis. It consists of 3 stages. عندنا 3 مراحل بمر بها تكسير الجلوكوز. Stage 1 known as the preparing stage. المرحلة الأولى تسمى المرحلة التحضيرية. Stage 2 cleavage of the fructose 1,6 bisphosphate. And stage 3 an energy generating stage اللي هي دي مرحلة إنتاج الطاقة. Stage 1 the preparing stage, it consists of 3 reactions: phosphorylation and isomerization and a second phosphorylation reactions. We mean by phosphorylation addition of phosphate group, and isomerization interconversion between 2 isomers. This figure shows the phosphorylation reactions. Glucose, which comes from extracellular fluid, it passes through the cell membrane and undergoes phosphorylation at position 6 to give glucose 6-phosphate using ATP as phosphate donor. As we know, ATP refers to adenosine triphosphate. It donates 1 phosphate group to glucose and it becomes ADP. Glucose, when it accepts phosphate group, become glucose 6-phosphate and become incapable of living cell. This is known as trapping action. اللي هو زي الشرك بتاع الفار بيحصل الرات تراب بيحصل له trapping inside the cell. الفار لما ندخل الشرك ما بيقدر يغادر، كذلك glucose 6-phosphate when it undergoes phosphorylation become incapable of living cell. The main purpose of this trapping action or phosphorylation is trapping inside the cell and activation. Glucose as such is not active and it has no ability to enter any metabolic pathway. When it undergoes phosphorylation, it becomes active and it has ability to enter so many metabolic pathways. This figure shows the steps of glycolysis. It consists of 10 reactions. Reaction 1 already talked about it, as we said. Glucose, which comes from extracellular fluid, it first undergoes phosphorylation at position 6 to produce glucose 6-phosphate using ATP as phosphate donor. This reaction is carried out by enzyme known as hexokinase or glucokinase. الانزيم موجودة في أسفل السلايد according to the number of reaction. Glucose 6-phosphate is isomerized into fructose 6-phosphate by enzyme known as glucose phosphate isomerase. Fructose 6-phosphate undergoes another phosphorylation at position 1 using ATP again to produce fructose 1,6-bisphosphate. This reaction is accomplished by enzyme known as phosphofructokinase 1, and this is the key enzyme of glycolysis. Those three reactions represent the first stage, the preparing stage. In the second stage, fructose 1,6-bisphosphate undergoes cleavage into two trioses, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, by enzyme known as fructose diphosphate aldolase. The aldolase is split fructose 1,6-bisphosphate into two trioses and combine the two trioses into fructose 1,6-bisphosphate. بيشتغل في two direction. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate place at equilibrium. They are interconverted into each other by enzyme known as triose phosphate isomerase. Glyceraldehyde 3-phosphate is removed as soon as it is formed to be used in the next step. عشان كده أي dihydroxyacetone بتكون will be converted into glyceraldehyde 3-phosphate لأنه الانزيمز بتاعت الجلايكوليسيس بتتعرف على الجليسرالديهايد. عشان كده لازم ان احنا لما ندخل الجلايكوليسيس بندخل ب 2 molecules of glyceraldehyde لأنه each dihydroxyacetone phosphate is converted into dihydroxyacetone. Put this in your mind لأنه لما نيجي نعمل calculation للإنرجي هنضرب في 2. Glyceraldehyde 3-phosphate undergoes oxidation and phosphorylation. Oxidation, this is carried out by enzyme known as glyceraldehyde 3-phosphate dehydrogenase. This enzyme uses NAD+ as coenzyme to produce NADH. The oxidation means removal of hydrogen. فبيحصل removal of hydrogen from carbon 1. This hydrogen is taken by NAD+ to produce NADH. الconversion بتاع الجليسرالديهايد الالديهايد conversion of aldehyde into acid is known as oxidation. Also glyceraldehyde 3-phosphate undergoes phosphorylation by inorganic phosphate. فبيحصل addition of phosphate group into carbon 1 of glyceraldehyde to produce 1,3-bisphosphoglycerate. In this step, NADH produce and this through the respiratory chain produce 2.5 ATP. This is the first step in glycolysis which produce energy. In the second step, 1,3-bisphosphoglycerate undergoes dephosphorylation at position 1 by enzyme known as phosphoglycerate kinase. الكاينيزز قلنا بيتعاملوا مع ال ATP. One phosphate group is removed from carbon 1 of 1,3-bisphosphoglycerate. This phosphate is taken by ADP to produce ATP. And this is the first step in glycolysis which produce ATP at substrate level. In the next step, 3-phosphoglycerate is converted into 2-phosphoglycerate by enzyme known as phosphoglycerate mutase. the mutase enzyme transfers the phosphate group from carbon 3 into carbon 2. 2-phosphoglycerate is converted into phosphoenol pyruvate by enzyme known as enolase. الانولase بيتعامل مع الووتر. It remove hydroxyl group from carbon 3 and hydrogen hydrogen atom from carbon 2 in the form of water. And there is a double bond between carbon 2 and carbon 3. لما عملنا removal للwater بيحصل double bond between carbon 2 and carbon 3. In the last step, phosphoenol pyruvate is converted into stable pyruvate by enzyme known as pyruvate kinase. And there is the production of ATP again. فبيحصل removal of one phosphate group from carbon 2 of phosphoenol pyruvate taken by ADP to produce ATP. By this step, glycolysis is complete. Differences between hexokinase and glucokinase. Hexokinase is specific for all hexoses. من اسمه بيشتغل على كل الهكسوزس سواء كانت جلوكوز فركتوز whatever. Glucokinase is specific for glucose only. Gluco means glucose. Hexokinase found in all tissues, RBCs, brain, adipose tissue, whatever. Glucokinase found in liver and pancreas only. Hexokinase has high affinity, low Km for glucose. يعني ده بيشتغل على الجلوكوز حتى ولو كان في low concentration. Glucokinase has low affinity, high Km for glucose. ده ما بيشتغل إلا البلاد جلوكوز level يكون high. عشان كده هو under stimulation of insulin. When there is the high blood glucose level, this stimulate beta cell of pancreas to secrete insulin. This insulin activate glucokinase. Hexokinase supply glucose for the tissue even in the presence of low blood glucose concentration. ده المهمة بتاعته يعمل supply للجلوكوز حتى ولو كان في low concentration. بيعمل phosphorylation. Glucokinase remove glucose from the blood following a meal. ده بيشتغل بعد الوجبة لما البلد glucose level يكون high. This figure shows the steps of glycolysis. As we know, the end product of glycolysis is pyruvate. This pyruvate under anaerobic condition is converted into lactate by enzyme known as lactate dehydrogenase. The differences between pyruvate and lactate in the structure, pyruvate contain double bond at carbon 2. When it converted into lactate, this double bond is broken using the pre-existing NADH which comes from glycolysis. يعني بيستخدم ال 2 hydrogen to break the double bond. NADH which comes from glycolysis under aerobic condition is converted into produce 2.5 ATP through the respiratory chain, but under anaerobic condition is used by lactate dehydrogenase to convert pyruvate into lactate, and therefore it has no role in energy production. Glycolysis in RBCs. In erythrocyte, the first site in glycolysis for generation of ATP may be bypassed, leading to the formation of 2,3-bisphosphoglycerate, which is important in decreasing the affinity of hemoglobin for oxygen. This figure shows the normal step of glycolysis. The first step in glycolysis for production of ATP, in reaction of conversion of 1,3-bisphosphoglycerate into 3-phosphoglycerate by enzyme known as phosphoglycerate kinase. This step in RBCs may be bypassed. 1,3-bisphosphoglycerate is converted into 2,3-bisphosphoglycerate by enzyme known as bisphosphoglycerate mutase. The mutase enzyme transfers the phosphate group from position 1 to position 2. This 2,3-bisphosphoglycerate is very important in decreasing the affinity of hemoglobin for oxygen. The hemoglobin has high affinity for oxygen and the tissue need oxygen. This product compete with oxygen for hemoglobin and decrease its affinity and make hemoglobin to release oxygen for tissues. Then 2,3-bisphosphoglycerate is dephosphorylated at position 2 to produce 3-phosphoglycerate, and 3-phosphoglycerate is intermediate in the glycolysis, which is converted into 2 phosphoglycerates, then into phosphoenol pyruvates, then to pyruvate. This figure shows the consumption and production of ATP and NADH. In the first reaction of glycolysis, the conversion of glucose into glucose 6-phosphate, there is the consumption of ATP. Also, the conversion of fructose 6-phosphate into fructose 1,6-bisphosphate, there is a consumption of another molecule of ATP. Conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate, there is the production of NADH. The conversion of 1,3-bisphosphoglycerate into 3-phosphoglycerate, there is the production of ATP. And also the conversion of phosphoenol pyruvate into pyruvate, there is the production of ATP. The conversion of pyruvate into lactate by lactate dehydrogenase, there is the consumption of NADH. Summary of energy relationships in aerobic glycolysis. In a reaction which is catalyzed by glyceraldehyde 3-phosphate dehydrogenase, there is the production of NADH. This enzyme converts glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. In a step of glycolysis which cleaved fructose 1,6-bisphosphate into 2 trioses, each dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate. Therefore, we have 2 molecules of glyceraldehyde. Each one through this reaction produce 2 molecules of NADH. Each of them through the respiratory chain produce 2.5 ATP. Therefore, we have 5 ATP. In a reaction catalyzed by phosphoglycerate kinase, there is the production of energy at substrate level and 1 molecule, and we have 2 molecules of glyceraldehydes. Therefore, we have 2 molecules of ATP. In a reaction catalyzed by pyruvate kinase, there is also production of ATP at substrate level, and we have 2 molecules of ATP. There is a consumption of 2 molecules of ATP in reaction 1 and reaction 3. So the total is 5 plus 2, 7 plus 2, 9 minus 2 equal 7. Summary of energy relationships in anaerobic glycolysis. In a reaction catalyzed by phosphoglycerate kinase, there is the production of energy in the form of ATP, one molecule, and we have two molecules of glyceraldehydes, therefore we have two ATP. In a reaction catalyzed by pyruvate kinase, which catalyzes the last step in glycolysis, there is the production of ATP, and we have two molecules, therefore we have two ATP. The total is 4. And there is the consumption of ATP for reactions which catalyzed by hexokinase and phosphofructokinase 1. Therefore the net is 2, because 4 minus 2 equal 2. Under anaerobic glycolysis, the pre-existing NADH, which are produced by enzyme known as glyceraldehyde 3-phosphate dehydrogenase, it has no role in energy production, because this NADH is used by lactate dehydrogenase to convert pyruvate into lactate. Fate of pyruvate in glycolysis. Pyruvate can be converted into acetyl-CoA by pyruvate dehydrogenase complex, this under aerobic condition. Or it can be converted into lactate by lactate dehydrogenase under anaerobic condition. Also, it can be converted into alanine by transamination. References for this lecture include Harper's Illustrated Biochemistry, Stryer Biochemistry, Lippincott Illustrated Reviews Biochemistry, Usmali Kaplan. عندنا more references عندنا لينينجر ديفلين لو عايزين more details. النت كمان مفيد في بعض الأشياء، عندنا على قناة اليوتيوب في simplified videos ممكن تكون مفيدة. تم الكلام وربنا محمود، وله المحامد والعلا والجود، ثم الصلاة على النبي محمد، ما ناح قمري وأورق عود. This is all I can say in this lecture. I hope you understood. Thank you for listening, and you have a great day.